Hey guys, this is Mrs. Foy, and this is the second half of the Introduction to Metabolism lecture. I think where we left off is we were talking about exergonic and endergonic reactions, and we were talking about the fact that exergonic reactions are reactions where energy is released because the free energy of the reactants is greater than the free energy of the products. And so the delta G is going to be negative, and that means energy is released, it's spontaneous. And in an endergonic reaction, the reactant's free energy is going to be less than the products. The products' free energy is going to be more, and so energy needs to be put into the system for the reaction to go, and the delta G would be a positive. So in general, catabolic reactions, so remember that catabolic means breaking down. And so catabolic reactions are reactions where you take a large macromolecule and you break them down into the subunits, like a polysaccharide to a monosaccharide or a protein to amino acids. And they're mostly exergonic reactions, and there's going to be a negative delta G, and there's going to be more work that can be done from those reactions. Anabolic reactions, where you are building monomers into polymers, like proteins from amino acids or monosaccharides into polysaccharides, are mostly endergonic reactions. Cellular respiration, take a look at this. You've got a glucose molecule that you're breaking down into these smaller molecules, CO2 and water. So that's an indication that it's going to be an exergonic reaction. And sure enough, here we have a negative minus 686 kilocalories per mole of glucose. That is an exergonic reaction. That's the basis of cellular respiration. And the reverse of cellular respiration is photosynthesis. So you take these two smaller molecules, smaller subunits, and you're building them into a bigger molecule, glucose, and that takes an energy input, which in this case is the solar energy of the sun. So one of the things that we're going to see, this is a big picture kind of thing now, is that the cell is going to use catabolic reactions, where it's breaking down reactions, to drive anabolic reactions, where it has to build up molecules, because we're going to be breaking down molecules in digestion, and then we're going to be taking that energy to build molecules that we need to run our cells. And so in general, what we're going to be doing is we're going to be taking this energy, and we're going to use an intermediary to kind of run those reactions. So here we can see that this is an example of like a hydroelectric system where we have some potential energy and there's water flowing down, but then because it's a closed system, it's going to reach equilibrium. And remember, equilibrium is when you have this balance, and if you have a balance, then you're not going to have any potential to have any work done. So your delta G is going to be zero. In an open system, if you allow the greater potential of dammed up water to flow into another system, which flows into another system, you're never going to reach equilibrium, theoretically. And so you have this flow of multi-steps where you have this energy that's flowing. And that's very similar to what is going to happen with the cell when it uses catabolic reactions to drive anabolic reactions. So remember, the whole purpose of all this is that the cell needs to be able to do work. And the cell does basically three categories of work. So it's going to be, as I said, doing chemical work. So it's going to be taking the energy from breaking down big molecules into smaller ones to be able to then run anabolic or building chemical reactions. As we saw in the last chapter, it's going to be having to do work to transport materials from a low concentration to a high concentration, active transport, and the cell is going to need to move things, mechanical work like beating cilia, shortening muscles, dividing, moving chromosomes during cell division. So those are the three basic categories of cell work. And what we're going to see is that the molecule ATP is going to be what we say the energy intermediary. It's the go-between between these big catabolic reactions and then the three types of work that the cell needs to do. So we say it's an energy coupler. So it's going to take the energy from exergonic reactions and transfer them to endergonic reactions. So let's look at the ATP molecule. You can see here is adenine. That's a nitrogen base. We're going to see that same nitrogen base in the structure of DNA. We have ribose, which is a 5-carbon sugar. And then we have, here's an ester linkage here. We have 3 phosphate groups that are highly electronegative, and those are kind of bonded together. There is a lot of potential chemical energy in those bonds. When you hydrolyze ATP, you get inorganic phosphate, and then you have the adenine, the ribose, and 2 phosphates left, which is ADP, and that is an exergonic reaction. Energy is released. So this is a big picture kind of thing. What's going to happen is the energy from catabolic reactions, energy releasing reaction, is going to be used to take that 3rd phosphate group and bond it to the ADP to make ATP. I've got more potential energy in the ATP molecule. I use hydrolysis or hydrolysis to break that, and that energy that is released from that bond is then used to do work for the cell. It's kind of an intermediary, so it's going to be running endergonic process or doing something else that the cell needs to do. The way that ATP works is it usually works by phosphorylating. Now there are other molecules besides ATP that also do this, but ATP is kind of the main molecule that does this. So what's going to happen is that when ATP is hydrolyzed and that 3rd phosphate, that inorganic phosphate group comes off, it is phosphorylating another molecule, and it makes it unstable. It makes it more reactive, remember, less stable. So here's an example of an endergonic reaction that the cell would like to have happen. So glutamic acid and ammonia bonding together to make glutamine, but that is a endergonic reaction that requires an input of energy. And so what the cell does is the cell's going to phosphorylate glutamic acid, makes it unstable and therefore more likely to form with the ammonia. And so it is coupling an endergonic reaction with an exergonic reaction, and the net delta G is a negative 3.9 kilocalories per mole, and so that reaction, what we say, goes. So that's an example of how ATP is going to drive chemical work, but ATP can also drive mechanical work and transport work by also phosphorylating. So we already learned in the chapter on cell transport that in these membrane proteins in the cell, when the cell needs to do active transport, a lot of times the membrane protein will be phosphorylated. That phosphate group came from an ATP that was hydrolyzed. When the phosphate group bonds to the protein, the membrane protein, it causes a conformational change in the protein that then is going to transport these substances, these solutes, usually against the concentration gradient. And of course, the phosphate, the inorganic phosphate in the ATP is then recycled. But the cell can also phosphorylate when it wants to do mechanical work. And we're going to learn about this when we learn about how muscles contract. So here we have an example of ATP being phosphorylated, and it's going to phosphorylate this little, what we call a motor protein, that has a conformational shape change. And this little protein is then going to have a conformational shape, and it's going to basically kind of walk down another kind of a stabilizing protein, like part of the cytoskeleton in the cell. So this is the way organelles can move in the cell, and this is also the way that our muscles move. And I am going to, I don't think that I can run this while I'm running a video, so I will make sure that I put this video on Edmodo so that you can watch it. This is a little video of a motor protein. So that's the way that ATP can act as a energy intermediary between exergonic reaction and endergonic reactions. And now we're going to talk a little bit about enzymes. So here's the deal. There in order for chemical reactions to occur, even spontaneous ones, okay, even ones that are exergonic, there are bonds that have to be broken so that they can be reformed to make products. And sometimes those reactions would be spontaneous, but they would be too slow to be able to happen in a timely manner to be able to support life. And the energy needed to be able to kind of get the reaction going is called an activation energy. So we've talked a little bit about this before when we were talking about enzymes, but now we're just going to go over that, just go a little bit more in depth. So look, guys, here's an example of an endergonic reaction. Okay, so look, I know it's endergonic because my reactants here, my products actually have more energy here. And so what's going to happen is it's an endergonic reaction, but still I have to have some activation energy that needs to be put into the system to be able to have the products be formed. Here is an exergonic reaction, so energy is released because the products have less energy than the reactant, but still, even though it's spontaneous, it still has activation energy. So that's a really important point. Even though a chemical reaction is spontaneous, you still have to have activation energy because you're having to break bonds. So this is a great example of this. So here we have our reactants. A is bonded to B, C is bonded to D. And in order to have these chemical reactions, these chemical bonds be rearranged, the original bonds have to be broken. And that takes energy to do that. And that's called the transitional state, where these energy bonds are broken so that new bonds can be formed in the products. And that's called activation energy. So what happens is that enzymes lower activation energy. Okay, so here is an exergonic reaction. Can you see that, right? Because the products have less energy here. And so this is going to be less free energy. I'm sorry, it's going to be a negative delta G. Notice that the delta G is not affected by the enzyme. The enzyme is only lowering the activation energy here. So the red line is what happens with when you have an enzyme, and the black line is when you don't have an enzyme. And I'll show you what happens. So let's see, did I show? No, here's another one. So here is an endergonic reaction, and again, we still have an activation energy here. The delta G is not as affected, but again, the red line is the activation energy that is lowered because the enzymes are involved, and if it did not happen, you would have a greater activation energy. So what is going to happen is that we're going to see that in a cell, you have lots of larger molecules that are full of potential chemical energy that would like to be able to process to go spontaneously through their chemical reactions. But they exist in the body. They're not just spontaneously broken down because there's an activation energy that's there, and they cannot get over that activation energy without enzymes. So enzymes allow the cells to be in control of chemical reactions. Otherwise, you would have these exergonic reactions from these big molecules happening kind of willy-nilly in the cell, and you can't, that would not be conducive to life. So here's what's going to happen. So here we have our big protein molecule, our enzyme. Here's our substrate. And we've talked before about how really the only potentially powerful part of the enzyme is the active site. So what we're going to see is as the enzyme moves into the active site, and remember it is a lock and key kind of mechanism. It is a fit, it's very specific fit from the substrate to the active site in the enzyme. What actually happens is when the substrate comes in, it causes a conformational change that actually tightens down, forms some temporary bonds between the substrate and the enzyme that we call the enzyme substrate complex. And that induced fit basically puts some pressure, so to speak, on the bonds that are formed in the reactant so that they can lower their activation energy and be more likely to react. And that's exactly what we see here. So we have our substrates coming in, and we have our induced fit, we have our enzyme substrate complex that lowers the activation energy basically because it's a spatial thing. It is holding those reactants close together, basically like you come here, okay, let's react. And so you have a chemical reaction. The enzyme is left unchanged and ready to, that's actually one of the definitions of an enzyme, is that an enzyme is not involved in the reaction and it also leaves the reaction unchanged. So we've talked about this before, but it just bears repeating that when you look at the rate of an enzymatic reaction, you can increase that rate by increasing the concentration of the substrate, but only up to a certain point. Now, if you increase the number of enzymes, then you can basically increase until you run out of substrate. But if you increase the substrate, you're going to have a faster and faster reaction until all of the active sites of the enzymes are what we say saturated. And then the speed of the chemical reaction is at its maximum, and we actually call that a Vmax. It can't go any faster because all of the substrates are already saturating the active sites of the enzyme. Two other parameters that are going to be important with enzymes is temperature. So different enzymes are going to have their optimal temperature that is best for their optimal rates of chemical reactions. And so we have, for example, very different in heat lumping or thermophilic bacteria. You're going to have a higher enzyme temperature rate than you would for typical human. And we also have very specific optimal pHs depending on where the enzyme is active. So here we have an enzyme in the small intestine where it's going to be a more basic or alkaline environment, and in the pepsid enzyme in the stomach, for example, it's going to have a more acidic. So two other molecules that are also involved in enzymes that are also involved to kind of tailor make the enzyme's controllability of chemical reactions are molecules called cofactors and coenzymes. So cofactors would be a non-organic, here's a cofactor right here, would be a non-organic molecule that reacts with a protein, an enzyme that is inactive, and by binding to it makes it active. So a cofactor is needed for an enzyme to be active, and a cofactor is a molecule that acts with an enzyme, but it's non-organic, so it's not carbon-based. A coenzyme is an example of a molecule that is an organic molecule that bonds with an enzyme in order to be able to activate it. So here's an example of what we would call a holoenzyme. It's a whole enzyme because its cofactor is bonded with it, and now it is active. Interestingly, that a lot of vitamins are actually precursors to coenzymes. And so they are very, very important for metabolic reactions in the body. So we know that we've talked about the fact that these substrates bond with active sites on the enzymes. And there are two different ways that enzymes can be inhibited, two basic categories. So one way is that you would have this molecule that would kind of get itself into the active site and compete with the substrate in that way. Okay, that's called a competitive inhibitor. And then you have other inhibitors that don't compete for the active site. They have another bonding site on the enzyme that causes a conformational change that changes the shape of the active site so that the substrate cannot fit. You see in both of these examples, though, the enzyme is not going to be able to do its job with the inhibitor in place. So an allosteric regulation is a type of regulation where you have another molecule that binds to the non-active site that is going to enhance or inhibit the enzyme. Okay, so we just saw an example. So this would be an allosteric regulation here, in which case you have a molecule bonding to a non-active site and that inhibits it. But you can also have molecules that bond to a non-active site and enhance it. And that would be an example of cooperativity. So here we have an example of an enzyme. All right, this is an allosteric enzyme with 4 subunits, and here we have a molecule that would be an activator. So it is going to bond to the enzyme, and it is going to in a site other than the active site, and it is going to then activate or stabilize the active form of the enzyme. Here we have an allosteric enzyme that is going to bond with an inhibitor, and the inhibitor causes a conformational change that causes the enzyme to be inactive, so the active site is not going to be able to bond with the substrate. And here we have an example of cooperativity where we have a substrate that is going to bond to the enzyme, and it's going to allow actually better bonding of other substrates. So the last thing I want to talk about today is this idea of negative feedback. And this is one of a major themes in living systems, but I just kind of want to show you this in a metabolic reaction. Very often in living cells, you will have multi-step chemical reactions where you end up with some type of end product, your final product. Each one of these intermediate steps is going to have a specific enzyme that is going to catalyze that reaction. And very often, the product at the end of those multi-step chemical reactions is going to be the inhibitor for the initial enzyme in the pathway. And when you think about this, this is such a great way to be able to turn off and turn on chemical reactions. If the point of this chemical reaction is to make a bunch of this end product, which in this case is isoleucine, and you get enough of that going, the cell doesn't want to waste energy by continuing to crank out isoleucine when it has enough of it. So here isoleucine is going to be an inhibitor, in this case an allosteric inhibitor, non-competitive inhibitor. It's going to bind to the non-active site of this original enzyme, number 1, this threonine deaminase, and it is going to prevent it from bonding with the original initial substrate, which is threonine. So it kind of turns it on. And then you can see that when the isoleucine concentration would begin to fall off because it's blocked up here, right? Isoleucine's not going to be made if I'm blocking this enzyme. The isoleucine concentration would decrease, and more of these would then not be able to bond to this other allosteric site, and this enzyme would be activated again, and so more isoleucine would be made. So this is called negative feedback because it is a way of turning off or going in the opposite direction of what the initial process was doing. So I hope that that's been helpful for you. That's the end of the Introduction to Metabolism lecture, and I hope you learned something. Thanks.